On June 24th, 2021, Professor Grainne McAlonan will present a webinar to the AIMS-2-TRIALS consortium and guests, including members of the autism community, about our shiftability studies. An introductory document was circulated in advance. This video has been created to answer questions arising from that document and help those who are new to the topic to understand the webinar.
So I'm Professor Grainne McAlonan, and I'm a Professor of Translational Neuroscience, and I'm based at King's College London. I also have a clinical job in the National Service for Adults with Autism and ADHD in the Maudsley Hospital in London. So in AIMS-2-TRIALS, I have a number of roles, and today I'm going to talk about our studies in the AIMS-2-TRIALS Shiftability project, which I carry out along with Dr. Eileen Daly at King's College London. So in the AIMS-2-TRIALS Shiftability project, we're studying the brain chemistry of neurodiversity. We use MRI, EEG, and sensory tasks to look at how the brain responds to particular drugs which target specific brain signaling pathways. We hope this will help us understand brain chemistry in autism, and we hope it will provide us with more and better tailored choices for people with autism, for example, for mental health conditions. Because multiple genetic and environmental influences have been linked to autism, these interact to make the brain very diverse. This means that each person may have a unique brain chemical profile. We can learn about that profile from the way a person's brain responds to a drug which targets a particular chemical system. So we're examining three brain chemical systems: the 5-HT or serotonin system, the glutamate and GABA system, and those are the chemicals that control excitation and inhibition in the brain, and also the endocannabinoid system, which in the brain is a target of cannabis medications.
What is a shiftability study?
A shiftability study is our term for an early stage study that asks if a particular medication is capable of having an effect on brain mechanisms relevant to autism. Adult participants with and without autism attend our study centre for 2 or 3 visits. On a given visit, they're given either placebo, a dummy medication, or a single dose of an active drug. Neither they nor the researchers know whether they've been given the placebo or active drug, and that's called blinding. And we then measure the brain responses and compare results with and without the medication. And we also compare the responses of people with and without autism.
What can we learn from these studies?
Well, these studies show us that the brain response to drugs in people with autism is not always the same as neurotypicals. So this tells us that the autistic brain is chemically diverse. So if we understand the differences in brain chemical systems in autism, and we understand what those chemical systems do in terms of the regulation of mental health and cognition, then we might be able to better tailor medication which could help with individuals who have particular mental health conditions or other problems they'd like choices for.
Have other medications been discovered through shiftability studies?
Well, in fact, we're among the first to use this approach in autism or any other neurodevelopmental condition. And in the past, there's been a rather non-specific approach to trying out interventions for autistic people, almost a try it and see approach. So we think it's better to find out if an intervention targets a particular mechanism before embarking on large-scale clinical trials. So this is a very new technique, and to my knowledge, there have been no medications discovered in this way, but hopefully it can be taken up more widely, and not just in autism, but also in other neurodevelopmental conditions potentially, and in other mental health conditions.
That was a slip of the tongue. We know autism isn't a mental health condition.
Because this is an issue for psychiatry, that we often come across medications by sheer chance, and that's no longer good enough. We need an informed way to develop pharmacological options for people who want that choice. We have recently completed a shiftability study using arbaclofen, and we are writing up those results. And arbaclofen is now in clinical trial. We've also completed shiftability studies on cannabis medications, and there are now clinical trials of cannabis medications in autistic people, including in AIMS-2 trials.
How do shiftability studies lead to larger clinical trials?
The studies carried out in a small group of people, and if we see an effect on an important mechanism, then that gives us confidence that that medication might be worth looking at in larger scale studies. If you use a drug study like a shiftability study, you might find out, yes, this particular drug in this particular person acts in this chemical system and changes that mechanism, hopefully for the better. Therefore, we could move into a clinical trial and see what happens to more complicated outcomes. And it may also help us avoid wasting people's time in clinical trials because of heterogeneity. So if we're able to think about, does an individual respond? And that's a reflection of their particular etiological mechanisms, then that's better than just, having this all-comers approach to clinical trials where everybody's invited. All the mechanisms that are happening are probably different across the cohort, and therefore there's no way your drug's going to be probably reach that primary outcome criteria. So it stops people also being exposed to medication, which potentially all you're doing is exposing them to side effects.
Can you tell who might benefit from the drugs using this method?
That's one of the things we're looking at. So I'll present some data we've got from a visual task, which we can have an individual index of whether there's a shift in that processing measure. And we could, for example, say the people who make that shift are people most likely to respond. That's one way of looking at it. Or we could look at the baseline measure and say, actually, some of those people have got a baseline measure that's very similar to the neurotypical group. And there's not much point in putting them in a clinical trial if the drug affects this mechanism, but this mechanism is actually completely typical, so there's nothing to work on. And in some cases, in the typical neurotypicals, what we find is that the drug acts in an opposite way. So you don't want to be giving people who have the sort of typical response a drug that actually then potentially maybe even has a detrimental effect. We don't know, but that's kind of the thinking behind it.
Don't you lose potential participants if you have a placebo condition this early?
So actually, we don't use placebo in the same way as conventional clinical trials. And everyone gets both a placebo visit and an active drug visit. So that way we don't lose any of the cohort. So a person who joins our study is asked to attend on, say, 2 visits. On one visit, he or she gets placebo. On the other, he or she gets the active drug. And the participant and the researcher don't know whether placebo or active drug has been assigned. And the order is random across participants. So half the participants may have placebo on their first visit. The other half will have active drug. The next part of that question was why we don't just use a baseline which might be already available, in fact, through AIMS-2 trials. So the reason we use a placebo is that we can control for other possible influences on the result. The placebo means that we have a baseline to compare the response to drug, which is identical in all aspects except for the content of the capsule administered. So for example, sometimes people are more anxious on their first visit and less anxious on their second. So we don't want everyone getting exactly the same administration on their first visit, because then we might just be looking at effects of anxiety rather than effects of drug versus placebo.
Have you considered including tests of executive function in these studies?
Let me first remind everyone what executive function is. So executive function is an umbrella term which describes the ability to plan, to inhibit responses, and to make choices in a flexible manner. And this person has asked if it has been considered to add tests related to executive function in these studies. And the answer is yes, indeed, it has been considered. And in fact, we've implemented that. So our approach is that more complex behaviors, for example, adaptive behaviors or social behaviors, are built upon fundamental blocks of executive function and sensory processing. So if we can understand how brain chemicals regulate those fundamental components of behavior, then we have a better chance of developing interventions, both pharmacological and psychological, which are targeted to mechanisms and ultimately contribute to more complex function. So in the MRI protocols we use, we include executive function tests, for example, of attention or response inhibition. We're also very interested in sensory processing, and I will talk about some of the results that suggest there are differences in the way visual stimuli are processed in autistic individuals, and that those differences may depend upon the GABA chemical signaling system. And just as the person who posed the question suggests, we think that issues such as sensory processing and executive function are really important to the autistic community, and that's why we've incorporated those tests in our project.
Have you considered that autistic people might have unexpected side effects and considered mid and long-term follow-up?
So these are incredibly important points, and actually why we think these studies are so useful, because the drugs we use are selected because they might have beneficial effects for at least some people with autism. Also because we only use single doses at a time. So if anything, we think this is a safer approach than signing people up to longer periods of drug administration when little might be known about a drug. For example, if we're using 2 doses, we try to use the lowest dose first, so that if a participant does experience any problems on that visit, then we can avoid inviting them for the higher dose visit. We remain in very close contact with our participants to ensure that they're doing well during the study and also afterwards. And in fact, many of our participants have helped us out on multiple studies, and we're really grateful to them. We very carefully screen all our participants and ask about reactions regardless of whether they have autism or not. Even so, we can never completely rule out an unexpected effect, but our participants are very well informed and they know that we cannot fully exclude unexpected side effects. Having said that, the only unexpected effect we've ever had was with one participant who vomited after the study started. And it turned out that he was given a placebo, or the dummy pill, but he was allergic to the red food colouring. So now we've learnt, and we know to ask about food colouring as well. And the participant was actually really nice and even asked if he could come back for another study, but without the red food colouring.
One person suggested that it would be interesting to measure immunological responses as part of evaluating a new treatment.
And I agree it would. In fact, we now appreciate that the neurons in brain work together with the brain's immune cells, so-called microglia, to regulate brain signaling. So we might expect a change in one part of this tightly linked system to cause a change in another. In this study, although we're not directly looking at the brain's immunological response, our data does allow us to collect some markers which might be relevant, including perhaps one of the chemicals that's found in the brain's immune cells, and that chemical is called glutathione. We're interested in that because that chemical is involved in the production of glutamate and GABA, which are those chemicals responsible for excitation and inhibition that I mentioned earlier. What we might find is that a drug which causes a change in the levels of one of those chemicals might be associated with a change in a mechanism relevant to autism. For example, an executive function mechanism or a sensory mechanism. And that's what we're targeting in the rest of our studies. We're doing it because those chemicals are interrelated. Therefore, unlike previous studies which have looked at those chemicals in isolation, we're trying to get a whole picture. So the idea that you may not see a change in, say, one of those chemicals, but maybe in the others is important to us because they're still part of a linked system. And it gives us a fuller understanding of brain biology. So I would say that that's a really important point. And there is work on immunology being done in other parts of AIMS-2 trials, but it's not a direct focus of this particular project.
Thank you for watching. Contact us via communication@AIMS-2-TRIALS.eu and keep an eye out for our public webinar series, which is starting soon. You can sign up for email notifications via Rebrandly, that's rebrand.ly/A2T newsletter, or visit the AIMS-2 Trials website at aims2trials.eu.